So imagine you've done everything right. You know, you eat perfectly, you exercise daily,
Right.
you take your statins religiously, your bad LDL cholesterol is just virtually non-existent on your blood work. You are basically the absolute picture of cardiovascular health. And then
you suffer a massive heart attack anyway.
exactly. Why? I mean, why does that happen?
Well, it's probably the single most terrifying and frustrating question in modern cardiology right now. We actually have a name for it. It's called residual risk.
Residual risk. Okay.
Yeah, it's basically the danger that's left behind after we've cleared away all the usual suspects. I mean, statins and those newer PCSK9 inhibitors, they do a fantastic job of like sweeping the main streets.
Right. Getting rid of the LDL.
Exactly. But they completely miss the fires burning in the alleys, so to speak. Things like triglyceride-rich lipoproteins, systemic inflammation, and the actual physical structural remodeling of your blood vessels.
So we have these blockbuster drugs that are essentially only fighting half the battle.
Barely half, yeah.
Wow. Which I guess brings us to the mission of today's deep dive. We're unpacking a truly groundbreaking 2026 paper published in Cell Reports Medicine.
Right, by Shirong Zhu and a really massive team of researchers from Peking University.
Yeah, and we're going line by line through this research today because, well, they might have just found the invisible culprit behind all this residual risk we're talking about.
They did. It's this previously overlooked protein called FAM3D. FAM3D.
FAM3D.
Oh.
Okay, let's unpack this, because if I'm a medical detective, I wouldn't even know where to begin looking for a microscopic killer like this.
Right. Well, you start by casting the widest net possible. So the researchers turned to the UK Biobank, which is this incredibly rich open access database.
Oh, I've heard of that. Huge amounts.
Massive. They didn't just look at a few dozen patients. They analyzed the blood plasma of over 53,000 adults.
Yeah. 53,000.
Yeah, and they tracked their health outcomes for nearly 15 years.
Tracking that many people for a decade and a half is just a staggering amount of data to sift through.
It is, but the scale is exactly what makes the findings so robust. They were looking at nearly 3,000 different proteins floating in the blood, just searching for a pattern.
And FAM3D was the one that stood out.
Lit up like a beacon.
Yeah.
There was a really striking, undeniable correlation between elevated levels of this specific circulating protein and both the prevalence and the future incidence of coronary atherosclerosis.
Meaning clogged arteries.
Exactly. Clogged arteries.
But, I mean, casting a wide net inevitably pulls up a lot of bycatch, right? Like, how did they narrow it down to prove FAM3D wasn't just some random biological noise?
Right. Good question. So they ran their own highly controlled 6-year prospective cohort study. Back in 2012, they took blood samples from hundreds of completely healthy individuals.
Okay, so baseline healthy people.
Yep. They measured the baseline FAM3D levels in their blood, locked those numbers away, and then just waited. 6 years later, in 2018, they brought everyone back to see who had developed enhanced CIMT.
Okay, wait. Let's make CIMT visual for everyone listening. That stands for carotid intimamedia thickness.
Right. That's right.
So think about the plumbing under your kitchen sink. CIMT is basically a specialized ultrasound that measures the thickness of the inner walls of the major arteries in your neck.
Exactly.
If that inner wall is getting thicker, your biological pipe is rusting and starting to clog. It's like the absolute earliest physical warning sign of cardiovascular disease.
A perfect analogy. And the predictive power of this protein was just unnerving.
How so?
Well, for every single standard deviation increase in a person's baseline FAM3D, which is just a tiny bump of 3.45 nanograms per milliliter in their blood,
Okay.
their odds of developing that dangerously thickened arterial wall 6 years later jumped by 27%.
Wait, really? A 27% increase in risk for every tiny 3.45 nanogram bump?
Yep. The odds ratio is 1.27. It's a massive red flag.
That is massive.
Yeah.
But I have to push back here for a second. In science, correlation is not causation.
Yeah.
Just because FAM3D is standing at the scene of the crime holding what looks like a smoking gun, how do we know it actually pulled the trigger?
Right. Can it just be a harmless byproduct?
Yeah, exactly. Like a byproduct of the arteries getting sick.
That is the exact critical question that separates observational science from mechanistic biology. To prove this protein is actually causing the disease, you have to run experiments where you control every single variable.
Right.
And to do that, you need animal models.
Wait, but mice don't naturally get human-style heart attacks, do they? I mean, you don't see mice dropping dead of clogged arteries in the wild.
No, you definitely don't. So how do you even test this without it taking a lifetime? You have to force the pathology.
Wow. How?
The researchers used a specialized viral vector. It's called AAV8-PCSK9. It acts like a genetic switch. It specifically degrades the LDL receptors in the mouse's liver.
So it essentially strips away the mouse's natural ability to clear bad cholesterol from its blood.
Exactly. It mimics the genetics of a human with severe familial cholesterol issues.
So they give the mouse a human-like vulnerability.
Yes. Then they fed these vulnerable mice a high-fat diet for 12 straight weeks.
Let me guess.
Yeah.
Bad news for the mice.
Very bad news. As expected, the normal mice developed severe hyperlipidemia, dangerously high blood fat, and thick, life-threatening atherosclerotic plaques.
Okay, but how does FAM3D fit into this?
Well, then they ran the exact same protocol on a very special group of mice. These were genetically engineered global knockout mice bred to completely lack the FAM3D protein. They call them FAM3D-.
And what happened when the protein was just totally missing?
The difference was night and day. Without FAM3D, these knockout mice had dramatically lower total cholesterol, lower triglycerides, and significantly less plaque buildup in their arteries.
No way. Even on the high-fat diet.
Even on the exact same high-fat diet with the exact same genetic vulnerability. But without that single protein, the heart disease was basically halted. Causation was established.
Wow. Okay, so if we step back for a second and just recap this first part, residual risk remains this massive problem. FAM3D is highly elevated in humans with clogged arteries, and higher FAM3D strongly predicts thicker artery walls 6 years later. And to everyone listening, think about your own annual blood work. We track LDL and HDL so carefully, but what if a simple FAM3D test could tell you your risk of plaque buildup a half-decade before it happens?
It completely changes the paradigm.
It's a chilling thought, honestly. It makes you wonder, where is this stuff even coming from? My first guess would be the liver, right? That's the body's cholesterol factory.
Yeah, the liver is the most logical suspect. But when they mapped out exactly where the body was manufacturing this protein, it wasn't coming from the liver.
Wait, it wasn't?
No, it wasn't in the kidneys either, and it wasn't coming from the immune system's white blood cells. It was being massively overproduced in the intestinal epithelial cells.
The gut.
The lining of the gut, yes.
So the command center for this heart disease mechanism is sitting in the intestines.
It really appears that way. But, to definitively prove it, they had to be genetically surgical. They couldn't just rely on the global knockout mice.
What did they do?
They used an advanced genetic editing technique utilizing something called VIL1-Cre genetics to create a new type of mouse. This mouse was perfectly normal. It had the FAM3D protein functioning everywhere in its body.
Except the gut.
Right, except in one specific location. They deleted the gene exclusively in the intestinal lining. They called this the FAM3D mouse, intestinal knockout.
So they basically silenced the command center. Did fixing the gut fix the blood?
It did. The blood lipids just plummeted. And it wasn't just a general drop. It was a massive reduction in the most dangerous types of fat.
Like which ones?
The very low-density lipoproteins, the VLDLs, and the chylomicrons. This localized knockout proved beyond a shadow of a doubt that the gut was the primary driver of this massive lipid drop.
Okay, here's where it gets really interesting, because we have to talk mechanism here. This sounds a little bit like magic. How does a protein sitting in the lining of your intestines manage to flood your entire circulatory system with fat?
Well, it all comes down to microscopic logistics. Inside the gut, there is a specific transport mechanism called MTTP. That's microsomal triglyceride transfer protein.
MTTP, okay.
When FAM3D is present in the gut lining, it triggers a transcription factor called HNF4 alpha.
Okay, HNF4 alpha. That's a lot of letters.
Yeah, it is. But you can think of this transcription factor as a factory boss. When FAM3D shows up, the boss suddenly starts screaming at the assembly line to build more of these MTTP transporters. Interestingly, it didn't affect another related pathway called AC2 at all, just MTTP.
Okay, let's use a mnemonic for this because MTTP is a mouthful. Let's think of MTTP as the master transport truck packer.
I like that, master transport truck packer.
Right, because when you eat a greasy meal, your gut breaks down and absorbs those dietary fats. But fats are oils, and your blood is mostly water. They don't mix.
Exactly. The fat can't just float into your bloodstream on its own. It would just pool and separate like a bad salad dressing.
Right. So the gut has to package these fats into microscopic delivery trucks called chylomicrons. That's a type of triglyceride-rich lipoprotein.
Yes, exactly.
So FAM3D is essentially a rogue signal that forces the gut to build thousands of extra MTTP packers. And because you have more packers, Every single drop of dietary fat gets efficiently shoved into these chylomicron trucks, maximizing the amount of triglycerides flooding into your bloodstream.
It's a massive flooding effect. But what makes this mechanism so insidious isn't just the sheer volume of the delivery trucks, it's the biological specificity. Like I mentioned, FAM3D doesn't touch AC2. It doesn't touch other cholesterol pathways in the gut.
It ignores them completely.
Completely. It is entirely laser-focused on upregulating MTTP via that HNF4 alpha boss.
Wow. So let's do a quick recap of subtopic 2. FAM3D acts in the gut. It boosts MTTP via HNF4 alpha, and then MTTP overpackages fats into chylomicrons, completely flooding the blood with triglycerides.
That's the mechanism in a nutshell.
It really makes you rethink the phrase, "you are what you eat." I mean, to you listening, how often do you think about the microscopic packaging plants in your gut, deciding exactly how much of that high-fat meal enters your blood? It's more like you are what your microscopic packaging plants permit.
That's definitely a much more accurate way to look at it.
But wait, earlier you mentioned that residual risk wasn't just about fat in the blood. You said it was also about the physical, structural remodeling of the blood vessels themselves.
Yeah.
Are you saying FAM3D is working two totally different jobs?
I am.
Like, it's raising fat in the gut, but doing something completely unrelated out in the arteries.
Yes. And this is honestly where the research shifts from brilliant to just truly groundbreaking. They discovered a second stronghold for FAM3D.
Where?
The endothelial cells.
The endothelial layer. Okay, for those visualizing this, that is the incredibly delicate single-cell layer of paint that basically lines the absolute inside of every blood vessel in your body.
Correct. So to figure out what FAM3D was doing there, the researchers deployed another targeted virus. This one was called AAV9 Ty2 Sham3.
Okay, another mouthful of a virus.
Yeah, but what it did was surgically silence the FAM3D protein only in the blood vessels, leaving the gut's fat-packing factories perfectly intact.
Oh, wow. So separating the two locations.
Exactly. And the result was astonishing. The atherosclerotic plaques in the arteries shrank significantly, but the blood lipid levels, the cholesterol, didn't drop at all.
Wait, so they reversed the physical heart disease without actually lowering the cholesterol. How is that physically possible?
Right. To prove this lipid-independent mechanism, meaning a pathology that has absolutely nothing to do with fat, they used a physical stress test. They performed a partial carotid ligation in mice.
What does that mean, practically?
It means they partially tied off an artery to create a bottleneck. This causes disturbed, turbulent blood flow. And crucially, they did not feed these mice a high-fat diet.
So their cholesterol was completely normal.
Completely normal. But that physical turbulence alone caused massive plaque to dig up.
Because turbulent blood flow acts like sandpaper. Right.
Yeah.
It aggravates and damages that delicate inner endothelial lining.
Precisely. And when they knocked out FAM3D in that specific area, the plaque formation stopped cold. They realized that the FAM3D sitting in the inner layer of the artery was actually launching an attack on the layer directly beneath it.
Which is what?
The vascular smooth muscle cells, or VSMCs.
Okay, let me try an analogy for this because the architecture of a blood vessel is so fascinating. Imagine the blood vessel is a tunnel.
Okay.
The endothelial cells are the paint on the inner walls. But right behind that paint are the vascular smooth muscle cells, the VSMCs. Let's call them the structurally sound bricks holding the tunnel open. Normally, these bricks are contractile. They have elasticity, they pulse and flex to pump your blood.
Right. In a healthy state, they express specific cellular markers, things like alpha-actin, calponin, and SM22-alpha that maintain that rigid, contractile identity.
But when FAM3D gets involved, it acts like a rogue foreman. It starts yelling at these structural bricks to stop being bricks. It tells them to abandon their contractile identity, lose those markers you just mentioned, and become what scientists call de-differentiated.
Yes, de-differentiation. It's a key concept here.
Suddenly, instead of holding the tunnel open, the bricks start frantically dividing, migrating, and moving into the center of the tunnel, physically clogging it up from the inside out.
That structural collapse is exactly why plaque isn't just fat sticking to a pipe. It is your own cells forgetting their identity, mutating, and migrating inward to build the bulk of the blockage.
That is wild. So, quick recap of subtopic 3. FAM3D operates in blood vessels independently of fat. It causes smooth muscle cells to abandon their normal contractile job, and these confused cells migrate and physically build the atherosclerotic plaque.
Exactly.
So to our listener, if a drug only lowers your cholesterol, but ignores this physical tunnel clogging behavior of your cells, are you really fully protected?
Not at all. It's a terrifying blind spot in modern medicine, but it also presents a massive biological mystery.
How so?
Well, how is one single protein, FAM3D, pulling the strings in the gut to absorb fat and pulling the strings in the arteries to destroy smooth muscle cells? Like, how does it actually communicate with two completely different types of tissue?
Right, because proteins don't have brains. They don't magically tell a cell what to do. They have to physically dock with a receptor, like a key sliding into a lock to trigger a chemical reaction.
Exactly. So to find that keyhole, the researchers use highly advanced molecular tracking tools. They utilize nano-by-TG protein dissociation assays and Brett assays.
Wow, okay. Without getting too bogged down in the lab terminology, they essentially made the proteins glow, right?
Basically, yes.
Yeah.
This allowed them to literally watch in real time inside a living cell which molecules were physically touching each other.
Like tracking fireflies in the dark to see exactly where they land.
Beautifully put. And by watching the glow, they found the universal lock. FAM3D binds directly to a specific receptor called FPR1. FPR1.
Okay, FPR1. That receptor sits on the surface of both the intestinal gut cells and the vascular smooth muscle cells.
Yes, it does. Both of them.
Okay, so the FAM3D key slides into the FPR1 lock. What actually happens inside the house once the door opens?
Once it binds, it activates a very specific internal alarm system. It triggers two distinct signaling pathways, known as the G-alpha-I and G-alpha-Q pathways.
No, I noticed reading through the methodology, they were incredibly careful to point out what it didn't trigger. It ignored other major pathways entirely, right?
Yes, it completely ignored G-alpha-13 and beta-arrestin.
Plaque.
That level of specificity is vital. In drug development, you want a sniper rifle, not a grenade.
Right.
And to prove that these two specific pathways, G-alpha-I and G-alpha-Q, were the sole culprits, the researchers used specific chemical inhibitors. They used PTX to block G-alpha-I and a compound called YM25489D to block G-alpha-Q.
I picture this entire cellular process like a massive game of molecular dominoes. FAM3D is the finger that tips the very first domino, the FPR1 receptor. When that domino falls, it splits the chain into two separate paths, the G-alpha-I and G-alpha-Q lines. Those dominoes cascade down, eventually crashing into the cell's DNA, causing the gut to build more fat packers and the blood vessels to collapse.
That is exactly the mechanism. And what those chemical inhibitors do is essentially pull a single domino out of the middle of the chain. When the researchers did that, when they used PTX or the YM compound, the entire disease process stopped dead.
So FAM3D was completely neutralized.
Completely. It couldn't upregulate the MTTP fat packers in the gut, and it couldn't cause the smooth muscle cells to de-differentiate in the vessels.
It's just mesmerizing. Let's recap this fourth subtopic really quick. FAM3D binds to the FPR1 receptor. It activates G-alpha-I and G-alpha-Q pathways. The single lock and key mechanism controls both the gut lipid absorption and the vessel wall remodeling.
Nailed it.
It's amazing how a single molecular finger can trigger such complex diseases. Think about it. Does knowing the exact molecular domino effect change how you view your body's internal clockwork? It's not just a vague concept of getting sick. It's a highly specific chain of chemical dominoes falling.
And, well, if you know exactly which dominoes are falling, you can build a tool to stop them.
Which brings us to the climax of the paper. We have the culprit, we have the weapon, we have the exact molecular fingerprints. So what does this all mean? How do we cure it?
The researchers didn't just map the disease, they built a countermeasure. They developed two specialized monoclonal antibodies named MAB642 and MAB643.
Monoclonal antibodies. For anyone unfamiliar, these are essentially lab-made, precision-guided missiles. They're designed to seek out and bind to one single specific target in the body, like the antibodies your own immune system makes to fight a cold, but weaponized specifically against FAM3D.
That's right. They engineered these antibodies to have an incredibly high affinity for the FAM3D protein. They seek it out and neutralize it. And when they injected these custom antibodies into their highly atherosclerotic mice, the results were staggering.
Did the plaques shrink, like in vivo?
Dramatically. Total cholesterol plummeted. The dangerous triglycerides, especially those VLDL and chylomicron delivery trucks, were slashed. And the arterial plaques physically regressed. All of this was achieved without causing any overt organ injury to the animal.
Hold on, though. I have to raise a major dilemma here based on what we talked about. We actually already have a drug on the market that blocks MTTP, the master transport truck packer.
Yeah.
It's a pill called lamidipide.
Yes, lamidipine.
But cardiologists rarely use it because it has a catastrophic side effect. It causes terrible, sometimes fatal fatty liver disease.
Yeah, because if you systematically shut down the MTTP packers everywhere in the body, you stop the liver from exporting its fat. The fat just builds up inside the liver cells until the organ literally fails.
Right. So doesn't blocking FAM3D do the exact same dangerous thing? Aren't we just trading a heart attack for liver failure?
That is the multi-billion-dollar question, and it leads to the researchers' greatest aha moment of the entire study. Think back to what we discussed earlier about where FAM3D actually comes from.
It wasn't in the liver. It was heavily concentrated in the gut lining.
Precisely. FAM3D only regulates those MTTP transporters in the gut. It has absolutely no effect on the liver's natural ability to function. So when they treated the mice with these new FAM3D antibodies, the gut stopped absorbing all the dietary fat, but the liver kept humming along perfectly normally.
So the mice didn't get fatty liver disease?
Not at all. It is a brilliant biological workaround, a targeted strike that entirely bypasses the toxic side effects that have made older MTTP inhibitors like lamidipide so dangerous to use.
That is incredible. It's not just a new drug. It's a structural hack to a problem that has plagued cardiovascular medicine for over a decade.
And the elegance of the therapy doesn't stop there. By targeting FAM3D, these monoclonal antibodies deliver what the researchers call a tripartite benefit.
A 3-in-1 strike.
Exactly. A 3-in-1 strike against the entirety of residual cardiovascular risk.
Okay, let's break those 3 down because this is the holy grail of what statins miss.
Number 1, it lowers the triglyceride-rich lipoproteins by shutting down the overactive packaging plants in the gut. Number 2, it fundamentally stops the structural vascular remodeling by preventing those smooth muscle cells in your arteries from going rogue and clogging the tunnel.
And number 3.
And number 3, it reduces systemic inflammation. Because it turns out FAM3D also acts as a signal that recruits inflammatory neutrophils, white blood cells, to the plaque site.
3 massive birds, 1 incredibly precise molecular stone. Let's recap this final subtopic. 2 specific antibodies were developed against FAM3D. They successfully reduced plaque and lipids in mice. They avoid the fatty liver side effects of existing drugs, and they offer a 3-in-1 strike against residual risk.
That's the summary.
We are entering an era where we don't just use chemical sledgehammers, but precision sniper rifles like monoclonal antibodies to treat heart disease. How do you feel about the future of custom-tailored medicine? It's just wild to think about.
It is incredibly promising. We finally have a line of sight into the invisible risks that have been killing otherwise healthy patients.
We started this deep dive talking about a broken X-ray machine, basically a medical mystery where a patient does everything right but still suffers a heart attack. But now, thanks to this Peking University team, we can see the invisible.
We really can.
We can see how understanding just one protein connects our diet, our microscopic gut biology, and the physical structural integrity of our arterial walls into one cohesive, treatable story.
You know, I want to leave you with a final, somewhat provocative thought that wasn't the headline of the paper, but is hidden deep in the supplementary data.
Oh, I love the supplementary data. What did they find?
The researchers noted that FAM3D acts very differently depending on the environment. When the knockout mice were fed a normal, healthy diet, removing the protein didn't change their blood lipids much at all. The entire destructive cascade, the gut flooding, the arterial collapse only exploded into action under the stress of a high-fat diet.
Wait, so the diet is what actually arms the weapon?
That's the implication. If this protein is so heavily influenced by what we eat to trigger these pathological pathways, it raises a profound question. Will the ultimate treatment for residual risk be an expensive lab-made monoclonal antibody? Or is the true cure uncovering how our specific individual gut microbiomes and daily dietary choices interact with these proteins to switch them on or off in the first place?
Wow, that is a fascinating thread to pull on. It brilliantly bridges the gap between high-tech molecular biology and the everyday choices we make at the dinner table. It's empowering to know that while our cells have these complex domino effects waiting to fall, our lifestyle might just be the hand hovering over the first piece. Keep questioning the world around you, keep looking for those invisible connections, and we'll catch you on the next deep dive.